Hello everyone, welcome to the next session or the week 2 session of the Introduction to Developmental Biology course Problem Solving PMRF-NPTEL session. If you have already joined last week or have seen the previous video, you will know that the PMRF-NPTEL sessions are problem solving sessions in which the Prime Minister Research Fellow tries to interactively solve various assignment problems tries to interactively solve various assignment problems from the previous year's assignments relating to the course so that the students who are taking the particular course can better understand and solve the problems themselves pertaining to this week's assignment. So let me introduce myself. My name is Anuvab Chatterjee. I'm Prime Minister Research Fellow and PhD scholar under the supervision of Professor Jonaki Shane at the Brain Developmental Biology Lab, Department of Biological Sciences and Bioengineering, Indian Institute of Technology, Kanpur. As before, in this particular session, we will go through the assignment questions one by one, go through the solution slides and try to solve them together, and then answer the question and move on. This will not only help in the students' a better understanding of the week's content, but also will give you an idea how to approach the various questions and understand the concepts behind them. So at least we have someone who has joined. Raja, are you there? Can you just speak up once?
Yes, sir.
Yeah. Were you there last week, Raja?
Yes, sir. Yes, sir. I was there last week.
Okay, nice. So we are going to go ahead. So you would at least know how we went ahead last week. Advait, hello Advait. Were you there last week as well?
Hello?
Yeah, were you there last week as well, Advait?
Yes, sir, for Developmental Biology Lab.
Yeah, the problem solving session that I took on Sunday.
No, sir, that I was unable to attend.
Yeah, okay. So this is your first session. So how we go about this is we go through the particular questions of last week's assignments, try to look at the solution slides, better understand the concept behind the questions, and then answer them. So these are essentially interactive sessions that the NPTEL PMRF are offering together. And so the key concept is that you should interact and like try to answer the questions as we go through them. So let us move ahead and look at the first question. So in this particular week, we had looked at developmental genetics at various concepts of the various theories that they help develop and carve the particular field that developmental biology now is. And one of the main important questions regarding it and one of the major developments during early years was the germplasm theory. So the first question that we're going to deal with is regarding the germplasm theory. So the question reads, what is or are the main points of the germplasm theory? The options are: changes in somatic cells will influence the genetic materials of the gametes. Each cell receives only the genetic instructions it needs. Chromosomes of somatic cells undergo fragmentation. And finally, germ cells provide continuity between generations. So this is a square box question, that is, there can be more than one correct. I would encourage you to try and answer the questions, like just go to the comments and try guessing or like answering which of the options might be correct. So like you can just go ahead and type it in. Okay, so let us then go ahead and look at the solution slide before answering the questions. So the germplasm theory was a very important development during early 1900s, and its importance is celebrated only second to Darwin's theory of evolution. So what the theory essentially tells us is that the entire heritable information that is carried in a particular generation is carried by the germ cells to the next generation. And each somatic cell only receives particular determinants required to form the specific somatic lineage. That is, the germ cell essentially has every determinant, or now as we call them genes, that are required to form muscle, blood, and neural cells. The particular somatic cells that divide off from the germ cells will only receive particular determinants in order to only form muscles, in order to only form blood, and in order to only form neural cells. They won't receive entire information, but the entire information is carried by the germ cell to the next generation. This flow of information is from the flow of information is from the germ cell to the somatic cells and not the other way around. Thus, information does not flow from the somatic cells to the next generation or to the germ cells. This is encapsulated in the central dogma of molecular biology by Francis Crick as well, in which so I would like to ask this at least. I think someone of you must know what the central dogma of molecular biology is. Anyone would like to try? There is the problem in trying and not being able to answer. At least the concept will be cleared. You can at least go ahead.
Mainly central dogma of molecular biology is the process that is from DNA to RNA, RNA to protein, and then the phenotype. So
Yeah.
that is the basic central dogma.
And so can you tell me, like, do you have an idea how the germplasm theory falls in line with the central dogma?
Sir, I will think, but I'm not sure I will answer it correctly or.
You can try. You can just go ahead and try.
Yes, sir, I will think. I will think.
Okay, so should I give the answer, or is anyone else trying as well? Okay, so let me go ahead and tell you. So as Advait told us, the central dogma is correctly from DNA. The information flows to RNA, it flows to protein, and then we have the phenotype. But in another way, as Francis Crick said, that the information flows from DNA to protein and cannot flow back. Once the information has gone to the protein, it cannot affect the DNA or the genetic code. This is essentially what the germplasm theory also tells us. Once the information has flowed from the germ cells towards the another somatic cell, which has already inherited the determinants in order to form muscle, blood, or neural cells, the information cannot flow back from the muscle cells to the germ cells. The germ cell is independent of these. This also opposes the Lamarckian view of evolution. This also opposes the Lamarckian view of evolution, in which it was told that many characters are main inheritance of heritable characters are there. That is, the blood cells of one generation can also send information of learned characteristics to the next cells of the next generation. That is, blood cells of this generation can can transfer information to the blood cells of the next generation. But the germplasm theory tells us that it is not possible. Only the germ cells will carry the information. So Advait, are you like now okay with how the central dogma follows up on the germplasm theory? Yes, no, anything. Comments also will do. Hello?
Yes, sir.
Yeah, okay. So if we try to answer the question now, what are the main points of the germplasm theory? After reading, obviously, it is obvious that the germplasm theory tells us that each cell receives only the genetic instruction it needs. As we saw in the previous picture, from the germ cells, muscle cells only get information, only get the muscle determinants in order to form the muscle lineage. Similarly, neural determinants are given to the neural cells in order to form the neuronal lineage. Other informations are not traveling from the germ cells to these cells. And germ cells provide continuity between generations. So this was the first question that we dealt with. I guess everything is okay. So I'm going to go forward. The next question is, chromosome diminution leads to, and there are four options: loss of parts of any chromosome in any cell, fragmentation of sex chromosomes, loss of only sex chromosomes in somatic cells, and loss of only autosomes in germ cells. So this is a round answer question. So there is only one option that is correct. I would like you to go ahead and try answering. Like, just type down whichever option you feel is correct in the chat box. Shriyan says it is B, fragmentation of the sex chromosomes. Anyone else wants to answer? Okay. So let us move on, but I would like everyone to at least try answering the questions, because getting them wrong here does not have any implications further on. You will just understand how we go about things. So let us look at the solution slide. So first, let us look into what exactly is chromatin diminution. So in chromatic diminution, during cell division, a portion of the chromatin is eliminated, which results in smaller amounts of genetic materials in the daughter cells. This can be seen here in this diagram, in which we see two particular cells, and after they have gone one particular round of cell division from this stage to this stage, we see that the chromosome have actually become smaller. And further on, you can see that this nucleus has broken parts of chromosomes, and this one also, and these are the somatic lineages. But this particular P2 marked cell has the entire chromosome and is actually the germ cell, and this germ cell contains the entire information in it, or the entire chromatid intact in it, but the somatic lineages is breaking up. So if we follow up to this stage, you can see that the somatic lineages have completely broken down, and this is better shown in this picture, in which the germ cells have the entire chromatic structure intact, but the somatic lineages have this genomes completely broken up, and only part of them are inherited in particular lineages, and the other parts are eliminated. This is what is known as chromatin diminution. The germline cells The germline cells thus continue to maintain intact chromosomes, as we can see right here, which is essentially supporting the germplasm theory, which says that the germline cells are what continue with, which carry information from one generation to the other, and thus they carry the entire information. But the somatic lineages only get what information they are required to form the particular somatic cells, such as muscle cells only will get information particular to the muscle lineage, and thus we see chromatin diminution in them. This was shown by experiments done by Theodore Bovary in 1901 on the Ascaris worm. So these are all images from there. Thus, if you go on to answer the particular question, what exactly is chromosome diminution? It is loss of parts of any chromosome in any cells. It is not particularly the autosomes or the sex chromosomes, but any chromosomes that are present in any one of the cells. That is what is known as chromosome diminution. Are we clear with that? Just sending a chat message with yes will also do fine. Shiansh?
Yes, sir.
It's okay, right?
Yeah.
Okay. So, based on the previous two questions, we have a third question, which which tells us what were the conclusions of Bovary's experiments that support the germplasm theory. So we have just seen the Bovary's experiments of Ascaris, which dealt on chromosome diminution, and we have also looked at germplasm theory in the previous question. So both of them are together in this particular part. So what are the conclusion of Bovary's experiments that support the germplasm theory? The first option is chromosomes carry the genetic material. The second one is chromosome fragmentation during division is observed in somatic cells but not in germ cells. Chromosome fragmentation during division is observed in all kinds of cells. And finally, abnormal development of cells is due to fertilization of egg cells by more than one cell. So this is also a round type of question, so we have only one answer correct. So please go ahead and try and answer the question. Which of the conclusion of Bovary's experiment support the germplasm theory? So anyone want to try and answer the question? We have already seen Bovary's experiment in the last slide, which was that. So Shiansh says chromosomes carry the genetic material is the answer. Kavya says it is also a chromosomes carry the genetic material. Anyone else wants to try? Okay, so we go ahead. So what exactly were Bovary's experiments? Bovary experiments, as we have seen in this slide, showed that chromatin diminution occur, and somatic lineages have smaller and smaller parts of chromosomes in them, and the germline carries the entire chromosomes in the entireties and thus carry the entire information to the next generation. Thus, if we look at both of the slides, we understand that actually the answer is B. Chromosome fragmentation during division is observed in somatic cells but not in germ cells is the answer. So, germplasm theory tells us that germline cells carry the information and they carry the entire information, whereas somatic cells only get whatever information they need in order to form the somatic lineages. And thus, Bovary's experiments, which also show that the chromosome inherited by various somatic daughter cells are actually smaller than the germline chromatins, supports the germplasm theory. Thus, the answer is chromosome fragmentation during division is observed in somatic cells and not in germ cells. So, the chromosome is getting smaller and smaller in somatic cells themselves and not in germ cells, and thus this shows us that somatic cells are getting smaller parts of the entire information that is essentially carried by germ cells to the next generation, and it is thus it is not A but B. Is that okay, or it was too confusing to understand? Kavya, Shianch,
Okay, sir.
you understood what I was trying to tell you, right? It is
Yes.
not that chromosomes carry the genetic material that the germplasm theory tells us. It is that the entire information is carried by the germ cells to the next generation. That is what we have to show, and that is what Bovary showed, and is the second point, that the chromosome fragmentation during division is observed in somatic cells but not in germ cells. Anyone wants to add anything else? Then it's fine. Then we go ahead. The next question is that which of the following cell types are exceptions to the genomic equivalence rule? The options are intestinal mucosa, hepatocytes, B lymphocytes, and T lymphocytes. So the first thing we have to know is what is the genomic equivalence rule. Anyone wants to try and answer the question? What exactly is the genomic equivalence rule? Okay, so let me try and answer it. Genomic equivalence rule tells us that which saw chromosome diminution occur, but that only occurs in worms such as ascarids. In other organisms, diminution does not occur, and every one of our cells carry the entire information that is inherited by the germline. This is what exactly is genomic equivalence, that the chromosomes will be replicated in their entirety, and the genomic material will be same in all the cells, be it somatic or germline. But there are particular cell types which are exceptions, that is, they do not form the follow the genomic equivalence rule. They do not carry the exactly same genomes as the other cells, and that is what the question is asking: which of these cells are exceptions to the genomic equivalence rule, that is, they do not carry the entire genomic information as the other cells in the body. So options are intestinal mucosa, hepatocytes, B lymphocytes, and T lymphocytes. So anyone wants to try and answer which one of these must be the I do not follow the genomic equivalence rule?
Sir, explain again, sir.
Okay, that is very nice. So what I want to tell you is that, say in ascaris, as we saw, so we go back. In ascaris, as we saw, the germline contains the entire chromosome, and the somatic lineages, which are shown right here, the nucleus you can see is fragmented. That is because the chromosome gets smaller and smaller with each cell divisions, and particular somatic cell lines, say muscles, only get the part of the chromosome that are required to form muscles. But the germline contains the entire genome. But this only occurs in ascaris. In all other most of the other organisms, such as us, the muscle cell and the nerve cell has the same genome. So the number of chromosome or the chromosomal sequence or the genetic sequence is same. This is what is known as genomic equivalence. The cells do not have difference in their chromosomal makeup or the genetic sequence, but there is differential cell gene expression that causes them to be different. That one is muscle and one is nerve, but the DNA sequence is same in all of them. This is what is known as genomic equivalence. Is that okay, Kavya? Or should I?
It's
You got it,
yes,
right?
sir. It's okay, sir.
So now what the question is telling us is that it is fine with muscles and nerves or most of the cells of the body, that the DNA sequence is similar or same. But some cells are there which do not follow the genomic equivalence rule. That is, there is some changes in the genome or the chromosome that are occurring. So which of the cells are these? The options are intestinal mucosa, hepatocytes, B lymphocytes, and T lymphocytes. Yes, Kavya says C and D. Advait also. Okay. Advait says the answer are lymphocytes. Okay. Anyone else wants to try? Raja says maybe B, hepatocytes. So hepatocytes are liver cells. So Raja says maybe hepatocytes. Anyone else wants to try? Okay, so let us go ahead. So I would want to know if any one of you have taken an immunology course before. Has any one of you taken an immunology course before?
Yes, sir.
So Kavya says no. Someone says yes. I can see the name.
Sir, now I am currently I am studying it.
Advait says yes. Okay. So have you gone through a concept? So this is a very interesting question. Say, you know that our body can recognize various antigens that are present, which are like for like various foreign organisms. So there is like unlimited amount of diversity of foreign organisms or foreign objects that can enter our body. So say those are like bacteria, viruses, pollens, whatever. Those are like infinite number of variety that is available in the world. But our body can form antibodies, or the immune system is ready to react against most of these foreign organisms and form specific antibodies. That is, it can recognize the infinite amount of diversity that is present in the world with ease. But the body has only a limited genome. We know that the genome size is limited. So, how can the immune system produce antibodies which are specific to seemingly everything that the world can throw at us with a limited resource? You understand what I'm trying to say? On but that was too complicated. Yes, no, anything will do. You can even type it in the comments that you did not understand what I'm trying to say. No, sir, very nice. So what I'm trying to tell you is that you know that there are various antigens that are present. There are these can be bacteria, bacterial proteins, viral proteins that the body has never seen before. But the body can form antibodies against them, and antibodies are specific to the particular antigen. So anything that is present, like the unlimited variety of antigens that are present in the outside world, the body can produce an antibody against it. But the antibody has to be created from the sequence. So antibody is a protein. So we know the central dogma. It DNA, RNA, protein. So the DNA is limited. We have only these many chromosomes, this much DNA. So how can the immune system form so many antibodies that can recognize everything? So this is a particularly important question that gets answered by this. So this is the question. The immune system generates antibodies against every possible foreign antigen from a finite genome size. So how is this exactly possible? Because if we have a finite genome size, then there should be a finite number of proteins that we can form. That also means a finite number of antibodies. But how can then a finite number of antibodies recognize everything that is out there, which is actually happening? So how is this possible? This occurs by a phenomenon known as Vdj recombination. So what happens is that in the lymphocytes, the B and the T lymphocytes, there are particular regions in the genome known as variable regions, diversity regions, and junction regions. These three regions can recombine. So recombination occurs in these three regions. So we have like essentially there is an option. You have V1 to V4, four V regions, D1, D2, and J1 to J3, three J regions, and these can recombine in any manner that is possible, and then form a final antibody. And so we have a V3, D2, J1 antibody, and thus there are many options that are present, and the antibodies can choose which region they want to there is like a huge combinatorial diversity that can be created. So from here only we can see that one single sequence can create 4 into 2 into 3, that is 24 different antibodies can be created from this region only. And there are other things such as that the junction region, even when it recombines, it can change its sequence so that there is a further diversity that is introduced into the whole thing. Thus, Vdj recombination occurs in B and T lymphocytes to give rise to this diversity by DNA rearrangements and recombination. Pair of DNA segments are chosen, double-stranded breaks introduced by Vdj recombinases, and deletion of intervening DNA and ligation of the chosen segments. So essentially the in-between part that is not being chosen by the cell are completely eliminated from the genome. And thus we have this particular this third genome size that is only present in the genome now. And this will get translated to form the antigen receptor domain. Thus, genomes of T and B lymphocytes is different from other cells because randomly some particular part of the VDJ sequences are being eliminated, and thus they do not follow the genetic equivalence rule. I think this was a bit heavy, but you can go through the slides and then tell me if you understand or not. Yeah. Okay. Not okay.
Okay, sir.
Yeah, maybe going through this or like reading up once will help you understand, because this is a bit complicated. But essentially what happens is that you have four possible I say we have 100 possibility of variable region, 50 diversity regions, and 30 junction regions. So thus from a finite number of finite DNA sequence, due to that 50 into 100 into 30 combination that can be formed, you can essentially have a huge number of diversity that is introduced. And thus the answer to the question is B lymphocytes and T lymphocytes do not follow the genomic equivalence rule. Let me add something. So there is another cell type in our body that does not have the its genome equivalent to other cells in our body. It essentially does not have a genome at all. Can anyone guess which cell I am talking about?
Red blood cells, sir.
Yes, exactly. So red blood cells do not have a genome or a nucleus. So if that was also an option, we could have gone with it. So that is this question. Next, we go into we brush up on our molecular biology a bit and look into the structure of genes. So this question particularly is very important to anyone who is trying to go for higher education, because most of the interviews, you will be asked a question similar to this. So the question is, with respect to eukaryotic gene structure, which of the following is or are correct? The first question is, the 5' UTR is present in the first intron. The 5' UTR is present in the first exon. Transcriptional termination is always after the polyadenylation signal. And fourth is, polyadenylation happens after splicing. So this is a square box question. That is, we have multiple answers that are possible. I would like you to go and... So Shian says the answer is C. Transcription termination is always after the polyadenylation signal. Anyone else wants to try? And please, this is a multiple choice one. So we have another try, I think. Shian says C. Kavya says A and C. 5' UTR is present in the first intron, and transcription termination is always after the polyadenylation signal. Anyone else wants to try? So I would again try to encourage that trying to answer the question will not be of any consequence. You will just get the confidence to answer and try to think of on the question itself. So I would like everyone to at least try answering the questions. Okay, so no one else seems to be willing, so we will go ahead and look at the gene structure and gene structure itself. So this is a bit complicated, but this is something that is asked very often, to draw the structure of a eukaryotic gene or that of a mRNA. So this is something that I would request you to go and practice. So here we have a structure of a gene, which is here is beta globin, and the first thing we have is the promoter region, which consists of the Tata box and the -70 sequence. After that, we have a transcription initiation site. This particular portion is something we all recognize. We have an exon followed by intron, then another exon followed by intron, and the final exon. But one of the most important things that you have to understand is, if the exons are what is present in the mRNA, everything that is present in the mRNA comes from an exon. So you can relate back that if particular questions are asked, like, is the translational termination codon part of the mRNA, or it is part of the genome, or it is part of an exon or an intron? This is something that you must be able to answer. Obviously, the translational terminator codon is something that the mRNA has. Otherwise, the mRNA won't know when the translation has to stop, and thus it must be a part of the exon. Similarly, you have to know that in an mRNA there is something known as an open reading frame, which starts from the start codon AUG and ends at the stop codon. Everything inside the open reading frame that is given in this curly bracket is going to be translated. But there is part of the mRNA that is not translated or untranslated, depending if it is 5 prime or 5 prime to the open reading frame. It is known as the 5 prime UTR or the 5 prime untranslated region, or the 3 prime UTR or the 3 prime untranslated region. Further, the mRNA has other modifications that occur in it. The mRNA has a 5 prime cap, which is a G cap, guanosine cap, that is a special nucleotide that is introduced, and it also has a poly A tail, which has a like 200 to 50 adenosines added to the tail of the or 3 prime end of the mRNA, which is not present in the gene sequence. It is only present in the mRNA. And finally, if we have to see that there is also the poly A addition site, that is the poly A signal is present in the mRNA, and thus it must be part of an exon. So these are all the information that you can get from this particular diagram. But the most important thing that you have to keep in mind is everything present on the mRNA and is part of an exon. If you remember that, you can go back and recognize that yes, this particular thing will be part of the intron, or that particular thing will be part of the first exon, and everything like that. Is it okay? Should I go through it again, or have you understood the entire diagram? It is a bit complicated, but I would like request you to just look through it and tell me. Is there anything that you want me to repeat, that something you are not recognizing? This is something that I did not look at before. I want to.
Then what will the intron contain, sir?
So the intron. Okay, so you have not gone through molecular biology course then, I would guess. But introns are particular parts of the gene that are not translated. Translated means that are not present in the protein, but they are present in the gene, and these are removed when splicing occurs. So when the mRNA is being formed, transcribed, splicing is a process that occurs that removes introns, and these particular portions are then removed.
Okay.
These are present in the gene but not in the mRNA.
Okay, sir.
Okay, and one of the other things you have to remember is introns is something specific to eukaryotes only. Prokaryotes do not have introns. Okay, and what exactly introns do is, I thought you were asking that. So introns are various regulatory elements. So you know that there are these things called enhancers. That was also dealt with partly in the this week lectures. That enhancers help in increasing, decreasing, manipulating the rate of the transcriptional process, help in maintaining the stability of RNA pol 2, and these can be present either 5 prime to the gene, that is upstream, 3 prime to the gene, that is downstream, or can also be present in the gene itself in the intronic sequences. So the introns play major roles in introns play major regulatory roles, that they do not get actively transcribed, but they help in the transcriptional process. Okay, Kavya?
Yes, sir.
Okay. So you have any other question regarding this particular diagram, because this is important, and this is like a basic thing that we need to know in order to in further lectures we'll have more and more things that are getting added on top of it. Anyone else has anything to ask? You can just go ahead and ask, please. Okay, then we can go on. So let us look back at the question. What the question was: with respect to the eukaryotic gene structure, which of the following is correct? So the 5 prime UTR is present in the first exon. So 5 prime UTR, as I said, is something that is present in the mRNA. everything that is in the mRNA should be exon, and thus 5' UTR is present in the first exon, not intron. Because if it was an intron, then it won't be transcribed and the mRNA won't have it. And 5' UTR is only present in the mRNA, and thus it is present in the first exon. Second is the transcription termination is always after the polyadenylation signal. So you can see right here that the poly A signal is before the transcription termination, because poly A signal should be present in the mRNA so that the poly A tail can be added, and thus if the poly A tail is added means it was part of an exon, and obviously therefore before the transcriptional termination site. So everyone is clear on this? Yes, no, I will request you to just go through it. If you have any doubt, you can ask me right ahead. Go ahead and ask me. Okay. So at least when I'm ending up with the question, I would at least ask you to like have a thumbs up emoji that you are correct, okay with this, so that I can at least know that you are looking at the screen. So everyone is, yeah, I get a yes. Okay. Okay, so that was the first question regarding mol bio, and we have the second question now. Choose the correct statement. The statements are: the poly A tail is cleaved from the nascent transcript of RNA before translation initiation. Phosphorylation of residues at the C-terminal domain of RNA polymerase II is a key regulator of the transcription elongation process. Electrophoretic Hello? Am I audible again?
Yes, sir.
Yes, okay. So let me go back. So at least you are able to see the question, I would guess. So we are back. You can see the PowerPoint, everything here. Yes, no. Yes, okay. And Kavi has already started answering the question. It is B. Nice. Otherwise, also says B. Okay. So the question is, so you have Shians also says B. Okay. So let us look at the solution slide. So this complex with the RNA polymerase II, the various transcriptional factors, the TF2B, TF2E, TF2F, H, A, D, is known as. Anyone would like to try and answer this? The entire complex with RNA polymerase II, various transcriptional factors that assembles at the Tata blocks before transcription starts. Anyone would like to answer that? So this seems to not be working. What does it? This seems to not be working. Okay, so am I audible at least? Hello?
Yes, sir. It's audible.
Okay. So yes, can someone try and tell me which particular what should be that complex that we are looking at?
Transcription initiation factor.
Transcription initiation factor. It, very close. It is the transcription pre-initiation complex. So it is a complex instead of a particular protein factor, that is like a complex of various proteins. So it is a complex and it is a pre-initiation. It forms before initiation of transcription, and that is why it is known as transcription pre-initiation complex. So there are various things attached, various transcription factors, importantly D, B, E, F, J, A, the RNA pol 2, and the C-terminal domain of RNA pol 2 is captured by the TF2D, the transcription factor D. And this is the start, the initiation phase is now over, and transcription factor D, what it does now is phosphorylate the C-terminal domain of the RNA transcript, and this essentially is the start signal. So this, in this position, as in a race, the runner was ready and in position to run, and this was the start signal which allows it to now go ahead and start the run, and now the elongation phase will start. This is the particular signal, and thus every one of you are correct. Phosphorylation of residues at the C-terminal domain of RNA polymerase 2 is a key regulator of the transcription elongation process. Okay, we are fine with this?
Yes, sir.
Okay. So the next question. Among the techniques listed below, choose the one suitable for identifying specific nucleotides contacted by a DNA binding protein. We need to know the specific nucleotides or the sequence which to which a DNA binding protein binds to. The options are Gal4 UAS system, DNase protection assay, RNase protection assay, and gel shift assay. So anyone wants to try and answer this? Kavya says B, DNase protection assay. Anyone else? Okay. Shian says A, the Gal4 UAS system. Anyone else wants to try? Okay, so we will go ahead and look at the solution slide. So the technique that we are that was the answer and we are discussing is known as DNase protection assay. What happens in the DNase protection assay is we first take a strand of DNA and we label it, and this is often a radioactive level, and then we divide it into two pools. In the first pool, we add a DNase, and the DNase then cleaves the DNA and forms a band-like structure such as this. And this can be here seen in the actual gel also in the 1, 2, and 3 lanes. We have various bands that are present. When only the DNase is added to the DNA sequence. Now in the second pool, we add the protein that we are suspecting can be binding to the DNA, and because the particular protein has bound to the DNA, the DNase, which is also a protein, cannot access the particular sequence, and thus that particular sequence is protected from the DNase. And what essentially that means is that that particular bands where the protein is binding will be absent from the gel. So this is this part that we can see right here in the fourth lane. This is known as the footprint of the protein, and thus this technique is also known as the DNA footprinting. And from the bands we can ascertain what the sequence was, and thus we know where exactly the DNA binding protein had bound. So thus among the techniques listed below, Choose one identifying specific nucleotides contacted by DNA binding protein. The answer is that DNA is but DNA is protection assay or DNA footprinting, whatever. The say names are different, but the technique they are referring to are the same. So the GAL for US system triage is essentially a conditional knockout system that uses enhancers. It was not. This was a bit different from the one that we are looking at. Yeah. Okay.
Yeah.
Okay. Next we look into what is known as pioneer transcription factors. PBX is a pioneer transcription factor because the four options are: it activates transcription without requiring any other accessory factors. It recruits other transcription factors to the promoter. It binds to its enhancer even if the enhancer is on repressed chromatin. And it directly interacts with the C-terminal domain of RNA pol 2. So why is a PBX a pioneer transcription factor is being asked. And we have four options. Anyone wants to try? Advait says B. It recruits other transcription factors to the promoter. Kavya says D. It directly interacts with the C-terminal domain of RNA pol 2. Anyone else? Okay. So first thing you have to know is what exactly is a pioneer transcription factor. A pioneer transcription factors are those that can directly bind to a heterochromatin or a repressed chromatin where transcription has not yet started. And that is why they are known as pioneer. That is the first transcription factor that can go and open up the DNA so that other transcription factors can bind. So here we have an example. So model for the role of pioneer transcription factor PBX in aligning muscle specific transcription factor MyoD on DNA. What PBX does is it recognizes its by DNA binding site even within nucleosome rich chromatin, that is heterochromatin or silent chromatin, where ordinarily transcription does not occur. PBX probably binds to the transcriptional factors. MyoD complex with its factor E12 cofactor is able to bind to PBX and replace transcriptional inhibitors. So these are the inhibitors present which are being replaced by MyoD. And then the MyoD can go ahead and recruit other transcriptional activators and start, such as MEF3 and MEF2, and start the entire transcription process. So the important part here is PBX is known as the pioneer transcription factor because it can go ahead and bind to silent regions of the chromatin where other transcription factors cannot bind. And thus the answer is C, that it binds to its enhancer even if the enhancer is on a repressed chromatin. Is that okay? Anyone wants to add anything?
No, sir.
Okay. So the picture in picture mode has gone up and not working. It is not good. Okay. So we will continue like this. Okay, so that was it. So Kavya says this it is okay, and anyone else wants to add anything? Advait?
No, sir. Sir, but it is partially also not correct na, that I have answered. It recruits other transcription factors to the promoter. So
Yes, you are correct that it also recruits other transcription factors, but that is something that other transcription factors such as MyoD can do also. The pioneer, the word pioneer is added only to a transcription factor when it goes and binds to repressed chromatin. MyoD or other transcription factors such as even TF2D, which is a normal, which is part of the pre-initiation complex, can also recruit other transcription factors. But the pioneer word is added only because PBS can bind to the repressed chromatin. You understanding what I am saying?
yes, sir. Yes, sir.
So this is the next question. DNA methyl transferase 1 recognizes dash on strands of DNA. Methyl guanine, methyl adenosine, methyl cytosine, or methyl thymine. C. Advait has gone ahead and answered C. Anyone else? Okay. So DNA Advait, you're definitely correct. The methylation of DNA occurs at the cytosine, and when methylation occurs at the fifth carbon, and thus we get the structure of 5-methyl cytosine. And this is what is recognized by the DNA methyl transferase 1. One other thing I want to add here is that there are two types of methyl transferase. One is the de novo methyl transferase, and the second is the perpetuating methyl transferase. De novo methyl transferases recognize CpG islands or CGG, C and G, a stretch of C's and G's that are present in the DNA sequence, and they go ahead and methylate a C that is present in the sequence. Once this C on one of the strands is methylated, then comes a perpetuating methyl transferase such as DNA methyl transferase 1 that was asked in the question, and upon recognizing cytosine with a methyl group added to it in one strand, it adds methyl group to the nearest cytosine in the other strand. And thus DNA methyl transferase 1 recognizes 5-methyl cytosine and then methylates a cytosine on the other strand. So Advait is correct. The answer is methyl cytosine. Anyone wants to ask anything or add anything to this? I guess not. Okay. So these the last questions are on genome imprinting. The question is, identify the correct statement or statements about genome imprinting. The options are: during gametogenesis, all existing methylations or DNA are removed and new sites are methylated. Second is, genome imprinting takes care of dosage compensation. Third option is, same mutation may cause different diseases depending on parental origin of mutation. And the final one is, new nucleotides methylated during gametogenesis differs between oocytes and sperm. So this is a square box question, that is more than one are correct. Anyone wants to go ahead and answer? Okay, so no, we have answers. Nice. So Raja says the answers are B and C. Genome imprinting takes care of dosage compensation, and C, same mutation may cause different diseases depending on parental origin of mutation. Kavya says A, C, and D, 3 options. So she also says that DNA methylation are removed and new sites are methylated, and new nucleotides are methylated during gametogenesis. Advait also says A, C, and D. And finally, Shriyan says A and C, that during gametogenesis, all existing methylation of DNA are removed and new sites are methylated, and C, same mutation may cause different diseases depending on parental origin of mutation. Okay, so let us look at what exactly genome imprinting is so that we can better answer the question. So what is genome imprinting? In case of many mammalian genes, the paternal and maternal chromosomes are differentially methylated in mutually exclusive genes. That is, there is a sex-specific DNA methylation. Depending if it is the sperm or the oocyte, the DNA methylation is different. And there is also the fact that only one allele of the gene is expressed. Depending on which parent the allele was obtained from, a severe to lethal mutation may occur. Say that there is a particular gene named A, and there is a paternal copy of the gene and the maternal copy of the gene. So normally what happens is, during genetic imprinting, the parental copy gets methylated. So the parental copy is already inactive. So what happens is, if the maternal copy is mutated, even though there is a perfectly fine paternal copy that the new embryo is inheriting, because the maternal copy is the one that should have been active and functional, and it is mutated, the whole gene's function is lost, and thus we have a severe to lethal phenotype that occurs. Is that okay, or that did not that was difficult to understand? You can tell me just it is okay, I understood, or it is very difficult.
Okay, understood.
Okay. And the last point is methylation occurs during spermatogenesis and oogenesis after removal of all methylation from the genome. So there is a reset button hit, and every methylation is removed, and then particular sites on the sperm or the oocyte is selectively sex specifically methylated again. So there is also another slide on this. This is an example of genetic imprinting, and this is the gene IGF2, which is genetically imprinted. This gene is activated by an enhancer element. It shares with the H19 gene, but that is irrelevant. We only want to look at this IGF2 only. So there is a differentially methylated region that is present between the enhancer and the IGF2, and there is a CTCF insulator protein, and this protein bind to silencer sequences and prevent transcription of the gene. So the RNA pol to the entire pre-initiation complex will form right here on the transcription initiation on the Tata box, which is around here. But due to the presence of this particular protein, it essentially acts as a physical barrier, so it can't move. So the CTCF blocks and silences the IGF2 gene, and this is present in both sperm and egg derived chromosomes. But in egg derived chromosome, the DMR is unmethylated and CTCF can just go and bind. But in the sperm derived chromosome, the DMR is methylated and the CTCF cannot bind due to the methylation, and hence the enhancer can and the entire pre-initiation complex can just go ahead and activate the transcription of IGF2, and thus only in the parental copy will IGF2 be active, and there will be an non-functional IGF2 in from the egg derived with the maternal copy. That is fine, okay, understood. You can even text in comment that yes, it is okay, not understood. Since no one is answering, I would guess that this was okay. So what are the answers? We have three answers that are correct. During gametogenesis, all existing methylation on DNA are removed and new sites are methylated. Same mutation may cause different diseases depending on parental origin of mutation, and new nucleotides methylated during gametogenesis differs between oocyte and sperm. Thus, three are correct. So, someone also said that B is correct. Genome imprinting takes case of dosage compensation, but dosage compensation is not something that is not entirely handled by genome imprinting. So, someone wants to tell me what exactly how dosage compensation is handled in embryo. Anyone has any idea what exactly is dosage compensation? Anything? Similar, no. So, what dosage compensation? Yes, someone was trying to answer. Okay. So, what dosage compensation essentially tells us is that in the females there are two X chromosomes, whereas in the males there is one X and one Y. So, does the number of genes that are expressed and the proteins translated in the females double that of males? Obviously not, because that will create severe phenotypes in females. So what happens is that dosage compensation occurs, which can occur in various ways. The most important that occurs is X inactivation, in which one X chromosome is randomly inactivated in the female, so that only one chromosome can be transcribed, and it is one that is expressed, and the other is completely transcriptionally silent. That is what dosage compensation means. It is not very related to genome imprinting. Okay. So this is the last question. Which of the following is the reason why the same mutation causes both Angelman and Prader-Willi syndromes? The options are random inactivation of one of the X chromosomes in females, sex-specific DNA methylation, random selection of one of the two alleles for inactivation, and sex-specific histone methylation. Only one option is correct. Anyone wants to try and answer this? Advait says B. B, that is sex-specific DNA methylation or genetic imprinting. Okay. So let us look at the solution slide. So Advait was right. This is essentially an example of genetic imprinting. So we have two locus present in both the sperm and the egg, and depending if the particular copy of the allele or if the particular chromosome is coming from the sperm or the egg, these are differentially methylated, and thus in the maternal copy only these red genes are transcribed, whereas in the paternal copy only the blue genes are transcribed and everything else is silent. Thus, when in chromosome 15 this particular in chromosome 15 a particular deletion occurs, which so you can see that these particular genes such as NDN is active only in the paternal copy and not in the maternal copy. Similarly, the SNO RNAs are active in the paternal copy and not in the maternal copy. So if a deletion occurs in chromosome 15 which inactivates a particular, say this entire portion of the chromosome goes missing, the other chromosome cannot essentially provide the particular genes which are needed even though it has them, because they are completely silent due to methylation. So if this the left part or the 5' part of the PWS is completely absent, there is a deletion, then the AS gene won't be transcribed by the paternal copy as it is completely silent in it, and thus depending where the deletion is in the maternal or the paternal locus, we have either the Angelman syndrome or the Prader-Willi syndrome. And if there is no deletion, we have the wild type. And if the both of them are deleted, so one of the parents has Angelman syndrome, the other has one of the parents has deletion in deletion in the maternal copy, there is a deletion in the paternal copy, then the embryo will have a lethal mutation and it won't survive. Thus, these are the inheritance patterns of the Prader-Willi and the Angelman syndromes. A region in the long arm of the chromosome 15 contains genes whose absence causes both these syndromes. But the conditions are imprinted in reverse fashion. That is why if one is active, another is inactive. In Prader-Willi syndrome, the paternal genes are active. In Angelman syndrome, the maternal genes are active. And this entirely depends on what the mutation exactly is. Thus, the same mutation, depending on where it is occurring, in the maternal or the paternal copy, can give rise to either the Angelman syndrome or the Prader-Willi syndrome. And thus, which of the following is the reason why same mutation causes both Angelman and the Prader-Willi syndromes? The answer is sex-specific DNA methylation or genetic imprinting. Is that okay or no? That was too difficult to understand.
It was okay, sir.
It was okay. Okay. So that was all I had for today.
Sir,
Yes.
sir, I asking question. This is a MCQ question. Okay, so they are the three option has been correct. Only one option correct. That during the examination, only one answer should be selected. So how can you determine they are the two option? Because they are the CBT examination on the online computer based.
Yes, continue please. In the CBT examination.
Hello. Question ki एमसीक्यू क्वेश्चन मतलब सिर्फ एक ही आंसर हम सिलेक्ट करेंगे ना, तो कुछ में तो तीन तीन आंसर सिलेक्ट होते हैं। तो ये बात एग्जाम में तो हमें सिर्फ एक ही आंसर सिलेक्ट वहां पे करना होगा, कि तीन तीन वहां पे भी होगा।
So, Shiansh, you can just go ahead. So, depend if you take the quiz, you will be able to see. If I select A and then select B, then if the highlighting moves to B, then that is a single option correct. And if both of them are getting marked, then that is multiple option. So, particular questions will be multiple correct, in which all of them have to be correct ones have to be marked, and some of them will be single correct, only which only the correct one has to be marked. So you can just go ahead and like just click three options, and if you see that all of the three are marked, so A gets blued, B gets blued, C also gets blued, that means that is a multiple choice. And if only one gets blued, then that is a single choice. In the particular assignment portal that is present in NPTEL, the round answer, so you can see in this particular question there is a round answer type of thing. This is a single correct. But if we go ahead and see this one, just we can see this is a square. The square indicates a multiple choice. But many of the times it is also possible that in during the final examination they will tell us which exactly questions are multiple choice and which are single choice.
Okay, sir.
Okay, you understood, Shiansh?
Yeah.
Okay. So this was all I had for today. The references to the slides are mostly Introduction to Developmental Biology, the 9th edition. If it is not written in the references are not written in the bottom of the page, then that means it is from the book. But if it is from outside, the references are given. This is the my YouTube channel, and you can go ahead and watch this particular video and the previous ones, and every one of the videos that are going to be uploaded hence will be uploaded to the YouTube channel. And in case you want to revisit the discussions, or if you have missed the questions and want to look into what exactly was discussed that week, you can just go ahead and look into the YouTube channel. So if anything else is not going to be added, I would say this is where I would like to stop. So anyone wants to add anything else? I guess not. Okay then. This thank you for listening to, coming in and joining the discussion, and thank you for listening to the video. See you next week.